All right, we're going to get started. Welcome everybody to the BICCN Omics Workshop, Day 2. All of today's slides and other materials can be found at this URL, BICCN 2110. And if you haven't already, please sign up for Terra at app.terra.bio. To set up the billing, you can use the pre-workshop instructions and fill out the form, or you can paste your email that you use to register for Terra into the chat, and the TAs can add you to the billing project. That billing project is going to allow you to do the hands-on activities today. All right. So before we kick off our intro, I first want to just introduce myself. My name is Elizabeth Kiernan. I'm a science writer at the Data Sciences platform here at Broad. And I'm joined today by my colleagues Anton and Leyla to give you a little intro to NeMO data analysis in Terra and data visualization in single cell portal. Before we kick off our presentation and give you an overview of Terra, we just want to do this little icebreaker activity. So if you could, in the chat, tell us a little bit about you. Answer 2 questions. What kinds of analyses are you doing? And what programming languages are you comfortable with? And it's totally okay to have no programming language. That would be my answer if I were to put this into the chat. But we just want to get a sense of where everyone's coming at. It'll help us tailor the workshop in the future. Great, I see a lot of answers coming in. Okay, a lot of interest in single cell research and some epigenomics as well. And I'm seeing a lot of Python and R users in the audience. Great. Thank you all so much for filling out those questions. Okay. Please feel free to use the chat today to ask questions. We do have TAs on hand. And with that, I'm going to dive into our Terra overview so that you can learn more about Terra, which is a bioinformatics platform that allows you to access data, run analysis tools, and collaborate on the cloud. So what is Terra and why is it on the cloud? If you're trying to unlock the mysteries of human genetics, today there's a lot of data to work with. Vast amounts of genomic files as well as phenotypic data in medical records. Governments and other institutions have invested millions of dollars into generating data sets like 1,000 genomes, which now has over 3,000 participants. UK Biobank, which has 500,000 participants, and all of us with over 1 million research participants. And these computing advances mean it's possible to turn this data into meaningful insights, the kind that can really improve people's lives. But while this sounds wonderful in theory, there's so much data that you can imagine it's hard to, one, find the data you want, the particular data you need for your study, download and store the data so you can analyze it, interpret and analyze file formats, and share your data with collaborators. And analysis comes with its own challenges too. You can imagine that working with all this data requires large, expensive infrastructure investments to handle the times when you need a lot of processing power, even though most of the time you might not even need that computing power. And that means that anyone who can't afford that kind of infrastructure is locked out. Analysis tools and computing environments are complex and often custom built, and it's hard to find the right tools and set up the right computational environment. And this is especially true if you're not a programming expert. And so these complex custom analysis tools and environments lead to non-reproducible results. But fortunately, the cloud can help solve this problem. And to illustrate how the cloud can help solve it, I want to just give you a little example of what the traditional approach is and how that differs from the cloud approach to bioinformatics. So in the traditional approach, we bring data to the researcher. Data lives in a central repository, and each researcher at their local institution receives a copy of that data. So not true data sharing, it's data copying. This results in high storage costs at each infrastructure, and everybody has to have their own tools on their local infrastructure to analyze the data. So that leads to non-reproducible results. It also means that each of these local infrastructures have to have fixed and expensive compute resources. Again, some people can't afford that. And there are individual security implementations at each infrastructure. So your security is only as good as the weakest security anywhere where there are copies of the data. In contrast, the cloud approach looks more like this. Data rests in a central repository, and researchers can access and analyze the data together in a virtual environment. This results in true and immediate data sharing, and you only pay for a single storage instance that many people can access. The tools that we use in the cloud are shareable. They're developed in what's called Docker containers. That means that you can get reproducible results because your colleagues can use the exact same tools that you use to analyze the data. And this is cost-effective, elastic compute. When you're working in the cloud virtual computing can expand to meet your analysis needs, which means you only pay for what you use. And you can imagine situations where maybe you have a grant coming up, you need more compute power to quickly pivot from one project to another. The cloud allows you to expand your research. You're not in that fixed, limited memory approach that you get with that traditional bioinformatics setting. And lastly, you have centralized data security, which removes the responsibility from the individual researchers and enables comprehensive security fences. So this sounds great in theory, but clouds are hard to use out of the box. You have questions like, okay, how do I find data in the cloud, the data that I actually need? How do I set up a compute resource so that I can actually analyze the data? How do I find cloud compatible code and tools? And how do I collaborate with my colleagues and share all of these resources with them? And how do I do this in a secure way? And that's where Terra comes into play. Terra is a bioinformatics platform that's developed by the Broad Institute in collaboration with Microsoft and Verily. And it's essentially a web interface designed to do the heavy lifting and help move your analysis to the cloud. Terra has unique capabilities designed to support research. So there's a data library where you can access public and access controlled data sets right from the platform. There are workspaces where you can bring together data and tools into secure and shareable units. You can perform whole pipelines on your data in Terra by running workflows. These are workflows that can scale, and you can bring your own, or you can explore tools that have been contributed by the community. And you can interrogate data in real time with different interactive analysis tools like Jupyter Notebooks, RStudio, and Galaxy. And Terra is just a hub in a large cloud data ecosystem. So Terra allows you to essentially access the rest of the ecosystem. You can use portals that are tailored to specific audiences, like all of us, Molecular Oncology Almanac. You can choose to bring your own data, or you can import data from these other resources and repositories. You can bring your own tools, but you can also find tools from external repositories as well. And Terra is building towards a multi-cloud system. So currently it's built on the Google Cloud Platform, but details about Azure are coming soon. Terra is built secure to work with highly sensitive data. Centralized security means that all components in the ecosystem exist within a FISMA moderate environment. And this satisfies the security requirements for many controlled access data sets. It's FedRAMP authorized as a moderate impact system. And you can find Terra listed in the FedRAMP marketplace. There's also a 15-minute idle timeout for clinical researchers if you step away from your machine. All right, so with that, we're going to get started with our hands-on activities. Before I do, I just want to see if we have any questions from the audience. Okay, so before I kick off our live demo of the platform and our hands-on activities, I'm just going to navigate to this resource here, this Getting Started link. This is going to take me to terra.bio. I wanted to highlight this resource because there's a lot of very helpful information on this page. There's a community forum, a blog, These blogs highlight different tools and use cases that are being used on Terra at the moment. We have a lot of guest features that come out. So this is just a really great way to get involved with the Terra community and find out how you can maximize the use of the platform. When you come to terra.bio, you'll also notice the big login button. If you click on that, it'll take you to the actual Terra platform, app.terra.bio. And this is where I want everyone to navigate to now, and we're going to sign in together. I'm going to give you a live demo, and then we're going to kick off those hands-on activities. So I will do my best to tell you when to watch and when you can do the activities. When you come to app.terra.bio, you'll notice 3 cards here at the bottom to help get you started. You have the option to view workspaces. This will take you to a list of all of your workspaces in addition to public workspaces and featured workspaces. You also have the option to view examples. You can be taken to our showcase library, which has workspaces with tutorials and cost estimates and example data. We'll go more into that in a moment. And then you also can go directly to the Terra data library and start looking for cohorts that meet your needs. But now I want to draw your attention to the main menu of Terra, which is in the upper left of the Terra platform. It's that horizontal 3-line icon. Go ahead and select that and log into Terra if you haven't already. You're going to sign in with the email that you used to register for Terra. All right. Now you can feel free to just take a moment and watch me as I demo some of the features in Terra, and then we'll clone a workspace. We'll find and clone a workspace together. So in this main menu at the top, you'll notice a profile dropdown. This will allow you to go to your profile, see what groups you're a part of, access billing information, any cloud environments that you have. The next option is this workspaces. Again, this is where you can go in and see all of the workspaces that you have created or copied, as well as different public workspaces and featured workspaces. There's a library dropdown, and we're going to explore this in a moment, where you can access the data repository, again, the showcase section with those tutorial workspaces, and a workflows page that will allow you to choose certain GATK workflows and also search for other workflows. So I'll show you these 3 sections in just a moment. I want to call out our support dropdown here. We do often get questions about where to find support documentation. We have a support forum available, so you can go to this support dropdown and get access to our how-to guides. We also have tutorials and videos available. And I want to call out the community forum, where you can post questions, but also find the answers to previously posted questions. So it's a very helpful way to get answers that you're looking for. And of course, you have the option to contact us with specific questions as well. If you are currently doing COVID-19 research, I just want to call out also the COVID-19 data and tools. These are resources in Terra that might be helpful for your research needs. All right, so let's go ahead and navigate to the library section. Again, you can just watch me. I'm going to start on the showcase page. So this is our featured workspaces page, and these are links to different workspaces where you can try out different tools and data in Terra. These are designed to be actual tutorial workspaces with instructions and downsampled data to allow you to test the tools at very low cost, or at times just a few pennies. You can search through these different tutorial workspaces to find tools that might meet your needs. For example, if you're interested in bioconductor, you can search the term bioconductor and find workspaces that can showcase different bioconductor tools. There's also faceted searches here on the left that you can search by. And you can also search by different data sets that you're interested in. So for example, BICCN related data. All right. And then I also want to call out our getting started tutorials. So if you're brand new to Terra, these are a great place to try getting more experience with the different Terra features like workflows, notebooks, and data. Okay. I'm going to switch over really fast to the datasets page, where you have access to the Terra data library. These are different repositories. Some have public access data in them, some have managed access data in them. For the public access ones, you're welcome to go in and browse the data, design cohorts, and specifically choose a cohort to meet your needs and export it to Terra. And I'm going to scroll down here and just show you that from here, you can also access the NeMO data portal, so the Neuroscience Multi-Omic archive. We're going to be showing a demo of this archive today, and you're going to actually be importing data from here. So when we get to this step today, I'm going to come back to this data library to browse data. And you will too, if you're following along. All right. The last thing I'm going to show you is our workflows library. Again, this is where you can find different GATK4 best practices workflow, but you can also search external repositories for additional workflows that meet your needs. So 2 options are DocStore, which is a GA4GH compliant platform, and the Broad Methods repository. All right, so now I'm going to show you how to find the workspace we're going to be using for today, and we'll talk a little bit more about workspaces. So go ahead and follow along with me for this part. So we'll find this workspace together. I'm going to go back to my main menu, that icon in the upper left, and go to the workspaces option. The BICCN Omics workspace is a public workspace. So we're going to be specifically searching for it today. When you come to this My Workspaces or this Workspaces page, this My Workspaces section may take a few minutes to load, depending on how many workspaces you have. If you've never made a workspace in Terra, this section will be empty. There are 4 pages available on this Workspaces page. The My Workspaces tab here, a new and interesting section, the featured section. This is all of the showcase tutorial workspaces that I just pointed out to you, and a public section. So if you're following along, go to the public section and select that page. Then at the top of your workspaces page, there's a search workspaces bar. Go ahead and type BICCN into that bar, and it should return the public BICCN Omics workshop. So go ahead and find the workspace really fast, and then I'm going to start a little tour of the workspace. All right, so this is the BICCN Omics Workshop Tutorial Workspace. Like all workspaces, at the top of the workspace is the name of the workspace, as well as a billing project that supports the workspace. And if you're not an owner of the workspace, you'll also notice a read-only option to the right of the workspace name. Over on the right of your workspace, you'll also notice a widget called the Cloud Environment widget. This is where you can launch a virtual machine to use applications like Jupyter Notebooks or RStudio. We're going to get some practice with setting that up today. When you come to any workspace, you're going to land on the dashboard. And the dashboard is a place where you can describe the workspace, what it's for, what kinds of data it has in it. And for our tutorial workspaces, as well as our showcase workspaces, you will find several commonalities between them. There will be usually a flow diagram laying out what kinds of exercises are available in the workspace. You'll see links to step-by-step instructions. And if the workspace has accompanying demos, you can find that in the workspace dashboard as well. And usually our tutorial workspaces have some quick start instructions for you. So if you're more familiar with Terra, these instructions can help get you moving faster through the exercises in the workspace. Another feature I want to point out is to the right, you can find out workspace information like when it was created, who owns the workspace, and importantly, what the Google bucket ID is. So every time you create or copy a workspace in Terra, it is going to automatically generate a Google Cloud storage bucket that goes along with that workspace. We call that the workspace storage bucket. And that bucket will have a unique ID with this FC prefix on it. Every person's ID is going to be unique to their workspace. So we're going to go ahead and actually make a clone of this workspace, and you'll see when you make a clone or a copy of the workspace, your bucket ID will be different from the bucket ID I'm showing you here. So let me show you how to go ahead and copy or clone a workspace. To do this, we're going to go to the vertical 3-dot icon in the upper right of the platform and click on it. And that's going to give you a drop down, and you'll see the option at the top to clone the workspace. So go ahead and, if you're following along, find that vertical 3-dot icon and select clone. This is going to open up a dialog box. You're going to be asked to give the workspace a unique name. We always recommend using your initials and the date to help you find the workspace later on. So today's date is 10th. All right. And then the description is populated from the workspace dashboard. So you, don't need to fill that in. Sorry, I skipped over this billing project portion. The billing project is either the billing project for today, if you're working on the workshop, or if you have another billing project, you can use that too. If you're on the temporary billing project for today, it's BICCN October 2021 temp, and the TAs will paste that in the chat. So go ahead and select that BICCN billing project if you're on it for today. Again, this description is populated from the workspace dashboard. And authorization domain is allows you to set up essentially a badge for the workspace to keep data private. We don't need to do that for today's workspace. So after you've filled in your unique name and a billing project, go ahead and select clone workspace. All right. So once you've cloned the workspace, you should see that you are now an owner of this workspace. I'm going to give you a little tour of the additional pages on this workspace now. So go ahead and just watch me. You don't need to follow along. And I'm going to start here on the data page. So the data page is a space where you can organize data into data tables. You'll notice 3 sections here, a tables section at the top, a reference data, and an other data. I'm going to go ahead and click into one of these data tables, this file data table. You can see that data tables are very much like spreadsheets. There are rows and there are columns, and each row is going to represent an entity that you're trying to describe with this data table. So this could be a sample, a participant, a specimen, a file. So each row is one entity, and then the columns can be used to put in metadata related to that entity. You'll notice that in this case, for this data table, there we have a sample ID in the first column. The first column of all of your data tables will always have an ID column. And we have links here to a couple of FASTQ files for this sample. If I select one of these links, you actually see that this FASTQ file lives in Google Cloud storage. We have the option to go to the Google Cloud platform and view this file, or I have the option to download it or move it around using a tool called GSUtils. That's Google's suite of tools for moving cloud data. And we can also host other information about this project, like sample IDs or project IDs. The reference data section is a place where you can load pre-generated reference files. And there are 2 options. These are both specific to human, but you can choose to do an HG38 or B37 human reference. And that's going to load all of the files that you need for doing genomic analysis in humans. So things like your FASTA files, reference files, dictionary, and index files. If you're not using one of those two references, there's an other data section where there's a workspace data table, and you can upload workspace level metadata, including alternative reference files, to this data table. So here you can see we have a couple of human and mouse reference files available in this table, as well as a couple of whitelists. We're going to be using this metadata when we set up our workflows today. And if you needed to add a new reference or string or variable that's applicable to your samples, you can just select this plus icon, add variable, and it lets you fill in the information. Just like our FASTQ files in the data table, these links for these different reference files link out to the reference files hosted in cloud storage. So again, if I click on one of these, you can see it's a file that actually lives in the cloud. All right, so now we're going to go to the notebooks page. This is where you can choose to launch a Jupyter notebook. This workspace has one notebook that we're going to be using for today's exercises, the Seurat notebook. I'm just going to show you this in preview mode here. If you're not familiar with Jupyter notebooks, they're essentially like a digital lab notebook. They have different types of fields or cells in them. They have markdown cells where you can describe the notebook and the types of analyses you're doing. And then they also have code blocks where you can run code in real time. So again, we're going to be launching this particular notebook today and running through it together. I'm going to go to the workflows page next. And here you can see that our workspace has two workflows that we'll be using today. This one, Optimus Mouse v2, and two, Cumulus. If I click into this workflow, I come to a setup page. We're going to be doing a couple of configurations today for our different workflows. On the setup page, you'll notice the title of the workflow, a version number for it, and the source of it. So again, workflows can be imported from the Broad Methods repository, but also from DocStore. DocStore interfaces with GitHub, so you can see this particular workflow lives in GitHub in the Warp repository. You can choose to configure workflows either by defining file paths or by using the data table. Today, we're going to be using data tables to configure it. And you'll notice there are steps to select data. You select the data table that you want to run, and then you can select specific data from that data table if you want to. And then here at the bottom of the workflow configuration page, you notice a couple of tabs. There's a script tab where you can actually view the WDL script that you're going to be running. For those that don't know, all the workflows in Terra are written in the workflow description language, also known as WDL. That's a human readable and writable coding language. And because Terra integrates with WDL, it pulls in all of the inputs from the workflow into a table so that you can specify the different strings or files that you need as input to the workflow. And similarly, it has an outputs tab. It can pull the outputs listed in the workflow into this table, and you can choose a particular table or path to write these outputs to. The last thing I want to show you is the job history page. The job history page allows you to monitor workflows after you've launched them. This workspace doesn't have a run on it, so we don't see anything here, but we're going to be looking at this today. All right, so let me go back to the dashboard and tell you a little bit about what we're going to be doing today and how we're going to be using this workspace. So today we are going to be finding a 10x data set from NeMO, specifically example MOP data, similar to that shown in the flagship paper. So we're going to show you how to go into NeMO, identify a cohort to export. We're going to export that data to Terra, specifically the FASTQ files for the data set. And then we're going to process those FASTQ files using the Optimus workflow. And so this is going to do pre-processing steps like alignment and metric, quality control metric, calculation. And it's ultimately going to produce a raw cell by gene count matrix. And we can use that count matrix as input to the Cumulus workflow, which will do some filtering, normalization, clustering on the data, and also produce some visualization outputs. And we can also import the raw cell by gene count matrix into a Seurat Jupyter notebook and perform more bespoke analyses using Seurat. So we'll go more into these different tools as we're using them today. Again, I want to call out that this workspace does have step-by-step instructions. So if you want to go and do these steps on your own after the workshop, you have instructions here to walk you through these different steps. All right, so we're going to get started by finding data in NeMO. And to kick us off, I'm going to turn this over to my colleague Anton, who will give us a little overview of NeMO and how to find data in NeMO using a faceted search. And then we'll get hands-on and find our own data for the day.
Hi, everyone. I'm Anton. I'm also from the user education team. And I'll be showing you how to use NeMO's faceted search capabilities to upload data to your Terra workspace. I'll begin with a little bit of background. You may have already heard some of this before, but if you missed any of the previous sessions, just for your information, NeMO stands for the Neuroscience Multi-Omic Data Archive, and it's a data repository for the NIH's Brain Research through Advancing Innovative Neurotechnologies initiative and some related brain initiatives. And it's built and maintained by the Institute for Genome Sciences at the University of Maryland School of Medicine. Data submitted to the NeMO archive is hosted both at the NeMO site and mirrored on the Google Cloud platform. And this includes both public and private data. So public data refers to data that can be freely distributed with the wider research community, and private data refers to data that is distributable only to a select group of users due to the nature of the data. So for example, human data with consent requirements needing data access approval from NIH. The University of Maryland School of Medicine has partnered with the Allen Institute's Brain Cell Data Center, the BCDC, and the Broad Institute to make it easy to work with the NeMO Google Cloud Portal data mirror within the Terra cloud. And NeMO is one of Terra's hosted data sets, so you can find a link to it through the Terra data library. So if you go onto the app.terra.bio website, you'll be able to find this link by scrolling down in the left-hand menu. And in today's demo, we'll be showing you how to find data through the NeMO data portal. So once you go to the NeMO data portal, you will first see this front page. In today's tutorial, we'll give you an introduction to using the NeMO data portal's faceted search capabilities. So you'll be able to carefully select data with very specific characteristics and export them to your Terra workspace for downstream analysis. So you can see here on the front page of the data portal a summary of all of the data that's hosted there. You can see at the bottom a summary of the number of currently hosted grants and samples and files. The data is also distributed across 4 different modality types, and you can learn a little bit more about the modality types yourself by mousing over each different color. So on this chart, the colors represent the different modality types, and those refer to different data motifs like epigenomics and transcriptomics. So this is a very versatile search because it allows you to build highly customized data sets, pulling in data from different cohorts, different data types, and sources. When you click on data on the portal's homepage to enter the faceted search, you'll see this page. The main portion in the center of the page provides a summary of the data you've filtered so far. In this screenshot, no filters have been added yet, so the summary includes the entire repository. You can see here all 700,000 files, all 5,000 samples, and also all the way on the right, you can see a convenient summary of the total size of the filtered data. So this includes all 180 terabytes. Above this is a search bar where you can manually input filters if you know the keywords for your desired filters. And on the left is a menu of some available filters. Note that both in the main section and in the filter menu on the left, there are multiple tabs, including both files and samples. Pay attention to this as you're going through the upcoming hands-on demo to make sure you're making the same selections as the presenter. In today's demo, we'll be selecting from some of the filters that are included by default in this left-hand menu. As you click on each category, you'll see a drop-down of the available options in that category, and you'll be able to click the checkboxes to add those options as filters. We'll be adding the mouse filter option under the organism category, the MOP filter under the brain subregion category, and the ATAC-seq filter under the techniques category. As you check the boxes, you'll see the filters populate to your filter search bar up here. Aside from those categories, there are a number of helpful categories included in the left-hand menu by default. For instance, you can also select on the basis of which team's sample sets you're most interested in by clicking on the grants category, where you'll see a list of teams associated with the brain cell data center. But you can also click Add Filter to add filters to the left-hand menu that aren't there by default. So once you've selected your filtered data, you will be able to see that data in what's called the shopping cart. And in this page, you'll see a summary of the data that you've selected. There will also be clickable items in your cart so that you can take a closer look at what each item is and remove them if you need. And of course, this is also where you can download the data either to your own local machine, or you can export the data to Terra. So under the download button, there's a little drop-down menu, and one of the options, which we'll be using in today's tutorial, is the export to Terra option. And once you click that, you'll be taken directly to the Terra website, where you'll either select an existing workspace where you want to send that data, or to start with a new workspace from scratch. Some helpful links for your information include a link to a wiki that describes the joint NeMO and Terra cloud computing environment, a link to the NeMO archives page containing links to the data portal, as well as to the NeMO analytics site, and a walkthrough that teaches you how to export the selected files and metadata from NeMO to the Terra data platform. So basically a walkthrough of the demo that you are viewing today. Now I'm going to pass it back over to Liz so she can take you through the hands-on demo.
All right, let's get started. I'm going to share my screen. Okay, so now we're going to get started with finding a unique cohort in NeMO and exporting that cohort to Terra. So if you're following along, go ahead and go to the main menu in Terra, that icon in the upper left. And we're going to find the NeMO data portal in the data library. So scroll down to the library dropdown, select it, and choose the data section. And once you're in the Terra data library, scroll down to the NeMO data portal. So I'm in NeMO, and then select Browse Data. All right. When you select the Browse Data, you're taken to the NeMO data portal, which should look very familiar based on Anton's presentation. You'll see a welcome to the NeMO data portal and a getting started by exploring. We're going to choose the data icon. And again, today we're specifically going to be looking for an example 10x V2 mouse single nucleus MOP dataset. So I'm going to walk you through how to do this faceted search. The TAs will be pasting the facets into the chat in case you want to follow along there. This might take a little bit of time to load because we're all loading it at the same time, just as a heads up. And of course, if you have any questions as we're doing this, please feel free to unmute yourself and ask, or you can chat the questions to the TAs. Okay. You'll notice on the left is where you can perform a faceted search, and you can search by both samples and by files. And then in the middle of the NeMO data portal is a graphical summary. And right now we can see that we're displaying all samples available to us, 5,017 samples. And as we narrow down the samples by using the faceted search, we'll see how this graphical summary changes. So I want everyone to go over to the faceted search side on the left. On the samples tab, go down to organism and select mouse. And you'll see how our different facets appear at the top of the, or in the top center of the portal. All right, and then our next facet will be under the brain region dropdown. So next to brain region, there's a little caret to the left. Click on that. And then you'll notice that we're looking for MOP, primary motor cortex, but it's not listed in the initial selections. So if you go down on the right of that facet, there's a 407 more. Click on that. And then I believe the MOP is about the 8th one down. So select MOP. And then go ahead and close that brain region drop down by clicking that caret again. We're going to go down now to the subspecimen types drop down. And this is because we want to specifically choose single nuclei data. So we're going to select the nuclei option. And then we're going to go under techniques and choose 10x chromium 3' V2 sequencing. So there's two options here, a V3 and V2, and we want the V2 sequencing. Okay, so now you can see that we have 15 samples, 72 files. Now we're going to go over to the files faceted search. So next to the samples tab, there's a files tab, and you'll see a format drop down. Go ahead and select FASTQ. That'll narrow down our files. Now we could choose to go ahead and add all of these files to our cart to export to Terra because all of these are MOP 10x FASTQ files. But in the interest of time today, I have a specific sample in mind that I want to export that has just 2 lanes of sequencing. So look at your graphical summary and go to the samples page next to the summary page. You should see samples 15. Click on that. And we are specifically going to look at the sample that starts with BICCN and it ends with the 328C. And the TAs will paste this into the chat. So to select this sample and add it to our shopping cart, we just click the shopping cart icon that's next to the sample name. And then you'll see in the dropdown that you can add all sample files to the cart or the filtered sample files. We want the filtered sample files. All right. Now that our samples are in the cart, I can see that if I go and look at the shopping cart in the upper right of the data portal. So I'm going to click on the cart. And if I scroll down, I can see the 2 samples that I have selected. We see we have a lane 1 and a lane 2 FASTQ file. To export these files to Terra, we're going to go to the download, the blue icon, blue download icon, and select that. And you notice we have a couple of options. We can download the file manifest and the sample metadata. Both of these are very useful when trying to understand how different data sets link back together, especially if you're choosing to export multiple samples. But we also see this export to Terra option. And what that option will do is export links to these files in their Google bucket locations and also the metadata along with those files. So if you're following along, go ahead and select Export to Terra. And that's going to redirect us to the Terra platform, where we'll be prompted to choose a destination workspace. And we want to start with an existing workspace because we're going to be using the clone that we just made. So choose the select or start with an existing workspace option. And then you can type the name of your workspace or choose it from the dropdown. I have quite a few files, so I'm going to go ahead and search by the date. Again, this is why it's useful to use the date to name your workspace. I'm going to select import. And then when I select import, it brings me back to my cloned workspace. All right, so let's go ahead and take a look at how our data page has changed now that we've brought in these samples. So we notice that we still have a file and file set table. And let's take a look at how those tables have changed. So in our file data table, we have a downsampled data that was preloaded into this data table before. But now you can see that we have our two samples that we just imported from NeMO. And if we go to the file set table, we now see that there are two sets, one again for our example data that was preloaded into this data table for us, and a new sample set that includes the files that we exported from Terra or from NeMO. Now, I want to emphasize that when you export data from NeMO, it automatically creates these data tables for you, and it names them file set. But if this doesn't match how you want to describe your tables, you can rename tables in Terra. And while NeMO chooses to export data to tables called file and file set, you'll find that other data repositories have their own names that they give data tables when you export data. You'll also notice we have two additional data tables. These are sample and sample set. This holds the sample information for the files that we just imported from NeMO. You'll notice the first column, like all columns in a Terra data table, is an ID column. It's a sample ID. And if I go back up to my files, I'll see that same sample ID listed in my data table. So here there's a sample ID column. So this sample ID will match the same sample ID in my sample data table. So you can link tables together using different IDs. And we have a sample set to represent all the samples that we brought in from NeMO. This time we only exported one library preparation with 2 lanes of sequencing. So we only have one sample in this data table. Okay. So let's go ahead and get started with analyzing the FASTQ files that we just imported from NeMO. So our goal today, again, is to analyze these FASTQ files using the Optimus workflow, which is a workflow that can takes in a single sample at a time, but it can handle multiple lanes of sequencing. So a couple of FASTQs per sample or library preparation. And what it's going to do is pre-process that data and produce a raw cell by gene count matrix in Loom file format. So what we're going to do today is select data to run with the workflow, and then we're going to write that output Loom back to the data table. Now, because Optimus often can take in an array of files, array of FASTQ files, again, multiple lanes of sequencing, I'm going to show you how to run this workflow on a set of samples. So here we are on the file data table, and you see the 2 entities that we brought in. Each one of these is a lane of sequencing and has their own FASTQ files. And then we have this set table, which groups those 2 lanes of sequencing together. So we have this file set ID and 2 entities here. So we could run our Optimus workflow on this data set, but in the interest of time today, I'm going to show you how to run on some downsampled data, which will run a little bit faster. All right, so to run on this downsampled MOP dataset, go to your file set data table and select the checkbox, next to the DS10M sample name. And again, the TAs will paste in the chat which sample you should select. So select the checkbox there. And when you select a checkbox, it's going to reveal a vertical 3-dot icon in blue at the top of the data table with the number of rows selected. Go ahead and select that icon, choose the open with option, and then we're going to choose a workflow to run on this data. And we're going to choose the first workflow for today, which is the Optimus mouse V2 workflow. All right. Now this workflow has been pre-configured for you with the exception of one input, which we'll go over. But anytime that you choose data from a data table to run with a workflow, it's a good idea to check your configuration. So I'm going to, again, confirm the name of my workflow and the version number. And because I opened data up using the data table, this is automatically selected for me. It says run workflows with inputs defined by data table. And it's going to, in step 1, ask you to choose a root entity type. That means choose the data table with the unit that you want to analyze. And so in this case, again, we want to analyze a set of files belonging to the same sample. We chose that data from the file set data table, and so this is automatically selected for us. But if we were manually setting this up, we would want to choose the file set data table. And then again, we already chose our data from the data table, so we have one file set selected. But if I wanted to choose a different data set to run on, I could do that by changing my setup here when I choose select data. But I'm going to leave it the way we have it because I want to run on that downsampled MOP data set. All right. When you have time, feel free to scroll through the inputs of the Optimus workflow. The required inputs will be in bold. The optional inputs will be italicized. I'll show you where you can find documentation on each of these inputs. But right now, I just want to show you how you can configure these inputs to pull from a data table. So we're going to focus on these first 3 inputs, and I'll show you the different ways you can pull data in. Now, remember, I told you earlier that you can pull files from the cloud by manually specifying a Google Cloud address or any cloud address for them. So I could type something like gs:// gs is the starting prefix for Google bucket URLs. And I could manually tell Terra where to find this file. But it's beneficial to use the data table because if you have to iterate through multiple samples when you're running in Terra, you don't want to have to go in and manually specify all of the URLs for those files. When the files are listed in a data table, Terra can iterate through them when they run a workflow. So that's the benefit of using the data table. It allows you to kind of scale your analyses more easily. This first variable that we're looking at is a variable called annotations GTF. It's looking for a GTF file to perform the analysis with Optimus, and it's specifically looking for a file. I'm going to just open up a new window to show you how these different data tables are pulled into the workflow configuration. So this is a variable called annotations GTF, and we actually listed this reference file in our workspace data table. So let me just show you that. Here's my workspace data table, and you can see I have a mouse annotations GTF file. Again, it lives in the cloud. So I can pull this reference file into a workflow by using the workspace dot syntax. I'm going to type workspace dot, and you'll notice when I do that, all of the reference files that are in that workspace data table are listed here. Let me just expand that. So here you can see all of the different options available. If I go back to my data page here, you can see those same options. So for this, I'm going to choose the annotations GTF. Oops, mouse annotations GTF. Okay. Similarly, I can pull strings from a data table into a workflow configuration. So this is an input ID variable from the workflow, and it's looking for a string name to demarcate this set. And so for this, I'm going to use my file set ID from my file set data tables. Let me just show you that file set data table. Here's an ID that marks the sample that I'm bringing in. Now to pull that, that ID is in my root entity data table. It's in that file set data table that I set to a root entity. So when you're trying to pull files from a root entity data table, you can use the this dot syntax. So I'm going to type in this dot. And again, you'll notice all of the options from that data table are listed. So all of the columns of this file set data table are listed when I type this dot. And again, I'm going to use that first column, the file set ID column, for this string. All right. Now, we need to specify a FASTQ file, a read1 FASTQ file for this workflow. And again, because Optimus can take in multiple lanes of sequencing, it's expecting an array. So before I can tell Terra which column has those FASTQ files in it, I first need to tell Terra what samples are part of the set that I've selected to run. So let me just show you what I mean by that. I'm going to go into the file set table here. And if I scroll over, you can see there's a files column that's part of this table. If I look at what this shows, it shows the different files from the file table that make up this set. So in this downsampled data set we have one file. In the data that we imported from NeMO just now, we have 2 files that are listed. So by telling Terra to look in this column, it will identify which samples are actually part of the set. So which files in this case. So the first thing I'm going to do is type in this dot, and then I want to type in the files column. Now that Terra knows where to find the files that are part of the set, I can tell it which column in my file table to go to find that FASTQ file. And that FASTQ file is going to be in this R1 FASTQ column. So I'm going to go ahead and type in R1 FASTQ. And then I'm going to select save. When I do that, you'll notice the orange icon disappears. So go ahead and do that yourself. Again, our TAs will paste this attribute into the chat so you can grab it if you weren't following along necessarily. All right. And then I'm going to go to my outputs page. So again, you can use this outputs table to write data back to a data table, specifically the root entity data table. And why would you want to do this? For 2 reasons. One, it allows you to search for outputs. So you can, when you go back to your data table, if you had a lot of samples that you were looking for, you could search for those samples or those output files. And it also allows you to chain workflows together if you write back to the data table, because you can use whatever you've put into the data table as output as input to another workflow. All right. So now we're going to be creating a new column in our data table for that output cell by gene count matrix. And again, it's in Loom format. So we need to configure this variable Loom output file to write to that root entity data table, the file set table. And so we can do that just as we did for the inputs. We can do that for the output using the this dot syntax, but we're not going to choose an existing column that's in there. We're going to create a brand new column for this output. And so I recommend using my Loom. You can call it pretty much anything you want, my output. So go ahead and type in a unique column name for your Loom file that you're going to write back, and then select save. And what this is going to do after we run this workflow is it's going to write back to this file set table a brand new column in alphabetical order called my Loom, and it'll contain a link to our Loom file. So it'll be right around here. All right. So if you've set up your inputs and your outputs, you're now ready to run the analysis. So go ahead and select Run Analysis and then Launch. And this will take you to your job history page. And we will come back and look more closely at this in a little bit. But now I want to go back into my workflows page to talk a little bit more about how to configure Optimus to meet your different research needs. Now, Optimus is a 10x pipeline that has been validated to run on both mouse and human. And it's also been validated to use V2 or V3 chemistry, as well as single cell or single nucleus data sets. So I just wanted to talk a little bit about what inputs you might want to change in order to configure this workflow. And then I'll show you some documentation on the dashboard that you can go to if you're interested in running this workflow on your own data. So the first thing is species. So we've already talked about how we have different species listed in the workspace data table. Let me just show you that again here. We have both human and mouse reference files available here. And so to change this from mouse, we would just need to human, we would just need to change the workspace attributes, these workspace dot attributes, to the different human reference files that are available in that workspace data table. The second attribute you can change is chemistry, and there's a couple of parameters that you need to change in order to accommodate. So right now it's set by default to V2 chemistry, but if you needed to change that, you would go to the chemistry attribute here and switch this to 10x V3. And you would also need to change your whitelist. So we are currently using a V2 chemistry whitelist. We need to change that to V3 to match barcodes. The other parameter that you can change is single cell and single nucleus. So you can do this by changing the counting mode parameter. So right now you can see this parameter is set for snRNA. We would change it to the scRNA. Okay. And so with that, we're going to take a little break, and then we're going to come back and I'll show you how to launch a cloud environment to get ready for our Jupyter Notebook analyses. And then we're going to take a look at the outputs of Optimus and how we can use those outputs as inputs to our Cumulus workflow. So let's go ahead and take 15 minutes. All right, welcome back, everybody. One question that I got in the chat while we were away was if I could show where the documentation for Optimus is. I realized I forgot to show that to you right before the break. So if you go to the dashboard of your BICCN Omics clone and scroll down to step 2, you'll see an overview of Optimus and a link to the Optimus overview in WARP documentation. So WARP is the Whittle Analysis Research Pipelines. It's the GitHub repository that hosts the Whittle script for Optimus. So I encourage you to go and check out this overview. It will give you all the details of the different inputs and outputs of the pipeline and configurations. And additionally, there is a featured workspace for the Optimus pipeline, which is in our showcase section, so you can check that out. All right. So now I'm going to go back and just show you the job history of our run. Let's see if it ran successfully, and it did. Now your run may not be done yet because you've never run this pipeline before. I have run this pipeline before, and so with the same inputs and outputs. So it runs a little bit faster if you use a feature called call caching, which I did. So mine's finished already. And so what I want to show you now is how our data page changes as a result of that run. Remember, I said we wanted to produce that raw cell by gene count matrix in Loom file format, and we wanted to write it back to our file set data table. So I'm going to go into this table now and scroll over, and we now see a new column called My Loom that has the output Loom file in it. And so we're going to actually use this Loom file as input to our Cumulus workflow, which I'm going to show you next. But before I do that, I also just wanted to show you in the other data section of the files, of the data page, you can go into files and you'll notice a UUID link in here. This is a folder that contains the execution directories for our workflow. So all of those outputs from the workflow are also written into each of these different tasks. So you can find outputs here as well. One thing I didn't show you earlier was that the files section of your data page actually is a link to the Google bucket associated with your workspace. So if I go in, say, and try to look at a file in here, you'll notice it says view this file in Google Cloud Storage. Let me just show you what that looks like really fast. You can see those same two files that I just saw in my Terra files section listed on the Google Cloud Platform side of this. So this is the Google Cloud Platform. We're no longer on the Terra platform, but the two are integrated. So that's why we can peek into this bucket from our Terra platform. We can also upload files directly to that cloud storage bucket. So we're going to talk a little bit more about the cloud storage bucket when we configure Cumulus in a moment. However, in the interest of time, we're going to first start launching our cloud environment. We're going to need to set up essentially a virtual computer in the cloud so that we can run Jupyter Notebook analyses a little later. So I'm going to show you how to set that up first before we launch our Cumulus workflow. If you have any questions about setting up the cloud environment, again, there's instructions in the step-by-step guide as well as on the dashboard. So here you can see which options you should pick when you're setting it up. We're going to use the default options, but we're going to make a slight modification to our CPUs. So let me walk you through that. So everybody go up to your cloud environment widget in the upper right and select the gear icon. This is giving you the option to set up a virtual computer. And to set up this virtual computer, we're going to use Docker containers, which are essentially portable packages of all the software and dependencies that you need to run certain applications in Terra. Terra has several default or pre-configured Docker containers for you. We're going to use the default one today, but we need to make a slight modification to it. So you'll notice this create custom environment option at the bottom of the dialog box. Go ahead and select customize. In the first part of the next dialog box, you'll notice an application configuration dropdown. This lists all of the pre-generated Docker containers available for you in Terra. So if you, for instance, if you're interested in using bioconductor, there's a bioconductor container with useful bioconductor packages preloaded. We also have some community maintained Docker containers for Jupyter Notebooks like Pegasus. And this section is also where you can choose to launch RStudio, so a Docker container with RStudio in it. You can also create your own custom environment. So if you don't see a Docker container here with the software that you need, you can create your own. And we have documentation in our support site on how to do that. So again, I highly encourage you to look into that. But again, for today, we're just going to use this default setting in our application configuration. In the second box here, you'll see Cloud Compute Profile. We're going to change the CPUs to 4. And when we do that, it'll automatically increase our memory to 15 GB. We don't need to change anything else. When you create these cloud computers, this cloud environment, it's automatically going to generate persistent disk storage for you. We don't need to change the amount of memory in this persistent disk. So let's go ahead and select create. When you choose create, you'll see the words creating now on your cloud environment widget. And like I said, it just takes a few minutes. So we're going to let that create while we run our Cumulus workflow. So the goal of our next section is to take that raw cell by gene count matrix that we produce with the Optimus workflow and use it as input to run the Cumulus workflow. Cumulus is a workflow developed by Bo Lee's group here at the Broad Institute. And it's going to perform filtering, normalization, differential expression analysis, clustering on that raw count matrix that we're going to use as input. So let's go back to our data page. And we're going to go to the file set data table. And once again, we want to use the Loom file that we just produced with Optimus as input to Cumulus. So I'm going to select that downsampled example data set again, the one that starts with DS10. And this time, I'm going to go to that vertical 3-dot icon in blue, click it, and select Open with, Workflow, and Cumulus. Now, I mentioned that we're using the Loom file as input to this workflow, but Cumulus can actually take in other file types like cell ranger outputs. So I encourage you to go to the Cumulus documentation to check out how you can customize this workflow to meet your needs. There are a few things that we've customized on this workflow. You'll notice that there are only 3 required inputs, but we have some additional inputs set. For instance, we're going to be using the Louvain algorithm for clustering today. We've also chosen to generate outputs that are compatible with single cell portal because Terra and single cell portal can integrate, and it allows us to visualize our data, and we can pull that in directly from our Terra workspace. So Leyla is going to be giving you an example of single cell portal later, but I just wanted to show you this option to generate SCP outputs. All right, so let's take a look at our required inputs. The first thing we need is an input file, and I already told you that we're going to be using our Loom file as input to this workflow. Now we could choose that to pull that Loom file from the column that we just created. However, if your workflow isn't done running yet, we have an alternative Loom file available in another column of that root entity data table, our file set table, called output Loom. So I'm just going to show you, I could choose to change this to this.myloom if I wanted to, but I'm going to go ahead and use the pre-generated one just so that we're all working with the same thing. This is pre-configured for you, so you don't actually need to touch it. The second thing we need to do in this workflow is specify an output directory. And this is where our Google bucket, our workspace Google bucket comes into play, because today that output directory that we want to specify is going to be in our Google bucket. So if you're following along, we need to find the ID for the Google buckets for our different workspaces. Remember, everybody has a unique one. So open your dashboard, right-click your dashboard and open it in a new tab. And remember, on your dashboard, the Google bucket ID is down on the right, and it starts with that FC. There's a clipboard icon right next to it. Go ahead and copy that. We're going to end up pasting it into our workflow configuration. Oops. So over here. Okay, let me move this over for a second. All right. So you'll see in this output directory, there's a paste bucket ID here. This is exactly where you want to paste your Google bucket ID. So what this is going to do is it's going to write a new folder to your Google bucket called Cumulus, and we'll show how that works after we launch the workflow when it's done running. The last variable we need is the output name. You could call this anything you want. This is a way to name your files. I'm just calling it downsample brain here, but you could choose a different name that's more meaningful to your data that you're running. And then I'm just going to click save to save the workflow setup here. I also just want to show you on the outputs, Cumulus is not configured to write outputs to the data model. Instead, it's going to write all those outputs to that output directory that we specified in our inputs. So if you go to the outputs tab, that's why you don't see anything pre-configured here, because we're not writing these outputs back to the data table. All right, when you're ready, you can select Run Analysis and Launch. And it'll take you to your job history. It takes about 10 minutes for Cumulus to run, so we'll check back on that. And we'll see how that changes our data page. And again, I just wanted to point out that there's documentation on Cumulus in the dashboard here. Cumulus also has a featured workspace, so you can check that out as well. And there's links to the Cumulus documentation here at the top in the Cumulus overview. All right. So while we're waiting for our Cumulus notebook, our Cumulus workflow to run, we're going to start using our Jupyter notebook, our Seurat Jupyter notebook, so that we can perform more bespoke analyses on that raw cell by gene count matrix that we produced with the Optimus workflow. So if you look at your cloud environment widget, you should see that it's running. And go ahead and select your notebooks page now and select the Seurat Jupyter notebook. Now, I showed you before that when you select a Jupyter notebook, it opens in preview mode, but in order to actually run code in this notebook, we need to open it in edit mode. So go ahead and select the edit page. All right. So the goal of this Jupyter notebook is to get you comfortable using Seurat to perform single cell analyses in Terra. And for those that don't know, Seurat is a suite of tools written in R for single cell analysis. This particular Jupyter notebook tutorial is a modification of a Seurat vignette for 10x PBMCs, but it's been modified to take in the MOP sample data that we're using today. That's featured in that flagship publication. Specifically, what this notebook is going to do today is we're going to show you how you can use code in this notebook to convert Loom files to a Seurat object. We're going to filter and visualize cells based on QC metrics, normalize single cell data, identify highly variable features, so genes. We're going to scale and perform linear dimensional reduction, cluster the cells, perform nonlinear dimension reduction, so make some UMaps, and then perform differential expression analysis and annotate our clusters. And my disclaimer here is just that the goal of this notebook is to showcase these analyses on Terra, not necessarily to teach single cell analysis. If you go back to the original Seurat vignette, there's a lot of rich details in there on these different tools. So I highly encourage you to check that out. There's a link to the original tutorial and the Seurat documentation here. The first part of this Jupyter notebook is a setup. We've already set up our cloud environment, but again, you can look at these notebooks in preview mode. So if you were ever looking at it, you could see the instructions for setting up your cloud environment so you could run it. And there are two ways that you can run a notebook in Terra. You can run all the cells at one time or run each of them individually. And I'm going to show you both ways. But before you run a notebook, we highly encourage you to clear the kernel. Because this will allow you to see what outputs are produced as you're running the different code blocks. So to clear the kernel, we're going to go to the top of our Jupyter notebook and select the kernel option. And then we're going to select restart and clear output. All right. Okay. So the first thing that this notebook does is it installs environment variables that make it easier for this cloud environment to communicate back with your Terra workspace. Because remember, we're actually running this notebook in a virtual computer. And so if we want to move files, say, back to the workspace Google bucket, we can do that by setting up some environment variables that allow this virtual computer to recognize the workspace. So I'm going to use this first code block to show you how to run code blocks in Terra. So you can click into this code block, and to run a cell or code block one at a time, you can select the run icon at the top here. You can also use a shorthand shift return. I'm going to go ahead and use that shift return. All right. And notice when I do that, a number appears next to it to indicate the order in which I ran this code block. All right, so what we're going to do is we're going to go ahead and run all the cells in this notebook, and then I'm going to walk you through what's happening in each of the code blocks as it's running, because it's going to take a little bit of time to run all of these blocks. So to run all the cells at once, we're going to go up to the cell at the top of the Jupyter notebook, select cell, and then run all. It's going to push you to the bottom of the notebook. Go ahead and scroll back up, and we'll walk through what this is doing. All right. So what our goal today is to show you how you could take that Loom output directly from Optimus, bring it into your cloud environment, and do some conversions on it to get it into a Seurat object that you can analyze. So the first thing after we set up our environment variables that we need to do is install our notebook packages. Now, this has been commented out because it takes a little while to do some of these conversions. But I'm going to walk you through it anyways, just so you could choose to run it if you want to. So if you wanted to do the conversion from Loom to Seurat object, you would need to install the SCEZ package. This is a special software that lives on GitHub, and there are instructions there as well on how to do this. So to run this, you would have to uncomment where it says uncomment the code below and run these code blocks. And the second thing you would have to do is actually move your Loom file into the cloud environment. Remember, your Loom file lives in your workspace Google bucket, and we would need to copy it into this virtual computer. And we would do that using GSUtil, which is, again, Google's suite of tools for moving cloud data. So there's instructions here on how you could find the Google bucket address for the Loom file going back to your data table, and where you would insert that Google bucket address. It would be insert cloud location. And then you could run this code per these instructions to move the file in. After moving your Loom file in, there's a two-step conversion that happens with SCEZ. So first you convert your Loom to ANData. And again, you would just follow the instructions and uncomment this code. This takes a little bit of time to do, about 10 minutes, which is why we're not running it today. And then you can check the files and make sure that you have that ANData file. It's an H5AD format file. And then the second step, you'll convert ANData to Seurat and produce a brain.rds file. And that brain.rds file, you could change the name on that if you wanted to a different file name by just writing over the brain part here. But this brain.rds file is what we're starting with for today's exercise. It's available in a public Google bucket. So the first thing we're going to do today is go ahead and move that pre-converted brain.rds file into our cloud environment. So here you can see that, again, we're using that GSUtil function, and we are bringing in this brain.rds file. And so when I list the files that are available in this virtual computer, you see that we have the brain.rds, and we also have this Jupyter notebook that we're running currently in the cloud environment. The next thing we need to do is load libraries that allow us to explore the single cell data in Seurat. And then we're going to read in the Seurat object using the read.rds function. And when we look at our variable brain, which is our Seurat object, we can see that we have an object of class Seurat with 55,536 genes across 127,35 samples and one assay, which is our RNA assay. So the first thing we can do is filter and visualize cells based on QC metrics. One nice thing about using the Optimus Loom is that Optimus produces quality control metrics that are commonly used, such as percent of mitochondrial reads and molecules. So you can read more about the Optimus metrics in the Optimus documentation in Warp. But it's nice because you don't have to regenerate those metrics necessarily. Seurat allows you to visualize metrics that you specify using the violin plot function. So here we're looking at that. And just remember that this is a downsampled example data set, MOP data set. So this is going to look a little bit like downsampled data. We can then filter the cells using a specific subset. In this case, we're using some suggestions from the Seurat vignette. We're going to subset our data set to features so that the cells express between 200 and 2,500 genes and have a percent mitochondrial molecules less than 5. And you can see when we look at the new object that we've subsetted, we now only have 8,218 samples. The next step is to normalize the single cell data. Seurat has a normalized data function that you can run to log normalize the data. And then we can identify highly variable features or genes using the find variable features function. And so here we're going to identify variable features and return 1,000 features per data set. And then we can look at the top 10 of those features and save it as variable top 10. We can make a plot of those variable features using the plot function. And when we do that, you can see the result is unlabeled. So we can label the plot using the label points. When we do that, we can actually see which features are highly variable in the data set. We can scale the data and perform linear dimensional reduction, so do some PCA analysis. To scale the data, we can use the scale data function and then run PCA using the run PCA function. And so this will return the genes that contribute to each of our principal components. There's a couple of ways you can visualize the PCA analysis using these 3 different functions listed here, and we can take a look at kind of how some of those look. So this visdim loadings, you can look at which genes are highly variable within this principal component 1 and 2. You can specify which components you want to look at using the DIMS specification here. We can also do a PCA plot. So here we're plotting PC1 versus PC2. And we can use a heat map to look at the different components. So here we're looking at principal component 1 and the genes that result from that. And we can also look at multiple principal heat maps for multiple principal components. So here we're looking at the first 15 principal components, and we can use these heat maps to kind of see where it looks like biological signal ends and noise begins. We can determine the dimensionality of the data set using an elbow plot. So Rod actually recommends a jack-straw plot, but in the interest of time, we're running the elbow plot. But again, I encourage you to go back to the Seurat tutorial to learn more about the jack-straw plot. So we can choose which principal components we think are contributing to the variability in our data set. And then we can cluster the cells using those principal components. Seurat uses the find neighbors and find clusters functions to perform clustering. And then we can do nonlinear dimensional reduction, produce a UMAP using the run UMAP function and specify those principal components that we want to use. So in this case, we're using the first 10. All right. And when we do that, we can plot the UMAP. And here you can see an unlabeled now UMAP with our different clusters. And at this point, we can save the file using the save RDS function. The next step is to perform differential expression analyses. Seurat uses the find markers or find all markers functions to do this. So in this first example, we're finding markers for cluster 4 using the find markers. And it's going to return a data table with those genes that are differentially expressed in cluster 4. And we have a little description of the different columns right below here, so you can go back and look at that. We can also use the find all markers function to look at the top genes differentially expressed in each of the clusters. So this takes a little bit more time to compute, but here you can see the cluster number and the different genes that identify that cluster. You can also change which statistical test you're using for differential expression. So an example is to use the ROC test. Again, you can read more about this in the Seurat documentation, but this will similarly return the differentially expressed genes. Now we can also visualize certain genes in the different clusters using violin plots. So I'm choosing to focus on some genes that were expressed in that cluster 4, so VIP and ERBB4. And so here we can see which clusters express those 2 genes. And we can also look at the top 10 genes expressed in each cluster and visualize that with a heat map. And we can use all of this information together to determine, to help annotate the different clusters. So I mentioned earlier that I was looking at cluster 4, and one of the genes highly expressed in that cluster was VIP. So VIP is commonly expressed in GABAergic neurons. So we're going to go ahead and, as an example in this data set, label cluster 4 for GABAergic neurons. And before we do that, we can use the feature plot function in Seurat to look at VIP expression in the clusters in a UMAP form. And then we can assign our GABAergic neurons to that cluster ID. So here I've left the other clusters numbered numerically, and then cluster 4 is given that GABAergic neuron ID. And then we can replot the UMAP with that annotation on it. So here we can see our GABAergic neurons. And the last thing we can do then is save this output back to our persistent disk location. And if we want to back up our work, we can move files from our virtual computer's persistent disk and move them back to our workspace Google bucket. So if you remember those environment variables we set up earlier on, they will allow us to move the files more easily just using the bucket variable. And then we can list the files that are in our workspace Google bucket. So here you can see all the files. So we have some runs from our Optimus pipeline, and now you can also see a Cumulus folder in there as well. So we're going to go back and look at that in just a second. But that's pretty much the conclusion of the Seurat tutorial. So at this point, you can feel free to go ahead. If your notebook is done running, you can pause your cloud environment. And we're going to go back and look at how the files changed on our data page. So I'm going to go into this files section on my data page and show you that here we are. Here's our different brain files that we brought in from our Seurat notebook. The other thing you'll notice is a Cumulus folder that we've now newly generated. If I go into my job history, you can see that my Cumulus workflow has completed running. And if I go in and look at those files, I can see the different files that are generated from the Cumulus workflow. So there's some differential expression analyses, some visualizations, a Loom and H5AD file. So let me just show you what one of these looks like. I can download one of them and open it up. And so here I'm looking at the differential expression analysis from Cumulus. And you can see it shows you the genes that are upregulated and downregulated in each cluster. So you can scroll through and take a look at the results from that. The other thing I want to point out are the single cell portal outputs that Cumulus produces. So any output on here that has this SCP demarcation can be imported into single cell portal and used to visualize this data. Okay. The last thing I want to show you in this workspace is how you can share all of this analysis with a collaborator. So when we're done running our workflows, we can go to the vertical 3-dot icon to the right here and choose the share option. And here I can specify a collaborator's email who's registered for Terra. And I can change their permissions. I can give them read, write, or ownership of the workspace. And you can read more about those different permissions in the Terra documentation. But it can get pretty granular for what you want to allow your collaborator to do. But essentially, when you give them permission to this workspace, they can come in and see all of your data and analyses. Okay, I think that completes this portion of the workshop. And now I'm going to hand this over to Leyla to talk a little bit more about single cell portal.
I'm going to go ahead and get started, and people can join as they come back from their break. So as Liz mentioned, today I'm going to tell you a little bit about the single cell portal, which is a web-based data portal for sharing and exploring single cell genomics data. And I'm going to do this in two parts. First, I'm going to show you some slides where I'll give you some background and walk you through some of the features of SCP that I think are the most cool and interesting. And then I'll also take you through a hands-on demo so that you can see how these features work in real life. So at this point, you might be wondering, what is the single cell portal? As I said before, it's a web-based data portal that you can use to explore and share single cell genomics data. But more than that, it is part of its value is that it's a centralized and standardized repository for this kind of data, which makes it a lot easier for people to discover your data and to work with it and analyze it if they want to, if it's interesting to them. In addition, it's designed to be a place where you can explore these data without requiring any computational experience whatsoever. Instead, we have a suite of dynamic, interactable visualizations that you can use to try to understand the patterns in these multidimensional data. And finally, SCP is designed to support all phases of the research lifecycle, from when you've just collected data and are sharing it with collaborators to try to suss out what the story, what story those data tell, to once you've figured out what that story is and you want to share it with the broader research community. And now is a really exciting time to be working with SCP because we're increasingly getting more and more data and now have a really rich set that come from researchers all over the world. And I think that you guys might find that there's a lot of value to accessing that richness, which, both gives you a broad audience for sharing your data and gives you the opportunity to discover some really cool data that are hard to find when you're just looking for them piecemeal. So as I said, I'm going to take you through a sort of highlights reel of the things that I think are the most useful and interesting when you're just starting to use the portal. So the first one is, once you get to the portal, you have to be able to identify the data that you actually want to look at. And for this, we have a few different kinds of search options, which I'll show you more in the demo. There's a basic search where you just type in some text, such as a disease or an organ, and that will search through the descriptions for all the studies on the site and return any studies that mention that term that you search for. There's also a more advanced faceted search that allows you to filter these results down based off of specific facets, such as cell type or anatomy. And this just makes it a little bit easier to narrow your search results so that it's more easy to wrangle all of those results. And in addition to being able to search across studies based off of this kind of terminology, you can also search for individual genes across studies. And I'll show you more about that in the demo as well. Next, perhaps the coolest part of SCP is the large suite of dynamic visualization tools that we offer to help you explore the data. So I'm going to take you through a few of these, though there are many different options, and I invite you to explore SCP to try to discover them all. So one really common kind of plot that you'll see for many studies is a scatter plot like this. This is the result of a dimensionality reduction that's been applied to a full data set, where that data set contains measured expression for a large set of genes in a large set of cells. So it's very high dimensional, but this has been projected into 2 dimensions in order to see the major patterns and the major axes. So in this kind of plot, each point corresponds to a cell or a bead, for example, depending upon how the data have been measured. And these points are all colored according to some sort of metadata that are included in the data, such as cell type or whether the sample came from a healthy organism or a diseased organism. And so, for example, you can explore different kinds of patterns by changing these labels around. And in some cases, the data are 3-dimensional, which is a really big benefit to be able to interact with 3-dimensional plots on the portal rather than in a manuscript, for example, because you can really appreciate the richness of the shape of the data when you can interact with it. Another really exciting and new type of visualization that we have is a way to look at spatial transcriptomics. So, for example, imagine that you've taken a slice of tissue like this, and you've put a matrix of beads on top of that slice in order to measure genetic expression at every location within the slice. Then once you've done that or measured something like cell type, you can then create a plot like this, which shows you it maps some information of the data, such as cell type, to every location on that slide and helps you understand the spatial distribution of the data. So when you look at this alongside a plot another dimensionality reduction plot like this that might be colored according to clusters that exist in the data, you could see interesting trends both in the data's expression values and in space. So for example, you might notice that this purple cluster is very separated in genetic expression space from the rest of the data. And it's also relatively segmented in anatomical spatial space, which might suggest something interesting about the mechanism behind that group of cells. I'll go through the remaining visualizations with less detail. We also have some violin plots that allow you to see the distribution of different genes expression in different segments of the data. There are also dot plots and heat maps that help you understand how different genes are related to each other in the data. There's also an option to create and save sort of annotations within the plots so that you can identify interesting subsets and share them. And finally, we have an ideogram viewer that helps you to search for an individual gene within a given study, and then quickly identify other genes that might be related to that gene, which might help you figure out your next target. So once you've been able to explore other people's visualizations, you might want to create your own study. And when doing so, you might want to be able to control who accesses your data and when. And so in anticipation of this, we tried to give you a lot of control over this aspect of the study. So first, SCP is included in Terra's security boundary, meaning that it's BICCN moderate, which gives it a layer of security. But even beyond this, we make it possible for you to control who gets to see your data based off of whether the study is a public study or a private study. In addition, when you share a private study or when you share any study with individual collaborators, you can control whether those collaborators can edit the study or just view it. And another really cool feature that we've just been working on is an easy to use system for anonymous reviewer access to your data. So if your study is private because you don't yet want to release it to the world until you've published it in a journal, but you want to be able to give access to the study to reviewers so that they can play around with the data themselves. You can use this system to basically send a pin to the journal and they'll pass that along to the reviewers, and then you can control how long they have access to your data, and they'll only be able to view your data without actually downloading it. But I'll tell you more about that in the demo as well. Another really cool feature of SCP, piggybacking on Liz's great presentation about running analyses in Terra, is that SCP is integrated with Terra, as I mentioned before. So you could put some data in Terra, run some batch analysis pipelines, or write some customized analyses using your notebooks. That might produce some standardized visualizations. And then you can publish those data and those results to SCP as a study. And then if you decided to sort of change your analysis along the way to create new results, you could sync those data between Terra and SCP. It just kind of reduces the barriers to sharing and analyzing data. Another way that we've tried to do this, which I won't go into as deeply, is to set up a few different ways to use scripts and APIs to access SCP's functionality, for example, to upload your data or to sync it. There's more documentation about this on the SCP website if you're interested. And finally, we've recently set up a system called study collections. These are kind of curated sets of studies within SCP that are united based off of the research consortium that's working on them, or the particular research topic. So for example, right now, we have a collection for the BICCN Anatomy and Morphology project, which is all of these studies are also available on the wider SCP, but it just allows you to sort of search through a subset that might be particularly useful for people that you collaborate with or with whom you want to share your work. we have another collection for COVID studies that were recently released. So as promised, that was a somewhat whirlwind tour of some of the highlights of SCP's features. I just want to mention before I transition to the next part of this presentation that I'm deeply grateful for the rest of my team, which is a dynamic group of designers, software engineers, and computational biologists, and to our funders for making this work possible. I'm going to throw up a few links to different resources, and I will pause here for a couple. I can take a couple of questions before we move on to the demo. Okay, well, I'll also pause after the demo, so that we're not done yet. Okay, can you all now see my the single cell portal screen? Great. Okay. Single cell portal. You can get there by just typing in this URL, singlecell.broad-institute.org/single_cell. You can also just Google it. That's how I normally do it. When you're at this homepage, I'm just going to first take you through a little bit of a tour of the things you might notice here. First, there's the search bar down here. So as I mentioned, it's easy to run a simple text-based search using this search bar. And I'll demonstrate how to do that in a moment, but I just want to draw your attention to the fact that you can search across for individual studies or through this gene search tool. Then at the top here on the right, there's a useful button for help and resources, which will link you to some of our documentation, as well as information on how to email us if you have any questions. And here's more documentation on using APIs and scripts to interact with SCP. Just to the right of that is this button to create a study. This is the first step in, you have to first sign in. So I will do that now, but you can see that I could also have signed in by clicking this button. But creating a study is basically just the first step in the process of uploading your data. At this point, you just name it, and you can point it towards an existing workspace on Terra or create a new one. And you can choose here whether you want to add it to a brand or a collection, such as the BICCN Anatomy and Morphology project, or just have it be a free-floating study. Okay, but I'm not going to create a study today. Instead, I'm going to start out by showing you some other features. So one last thing I want to point out on this homepage before we dig into some actual data is this button right here, which allows you to browse through the study collections. So I'm just going to briefly sign up, because otherwise I'll show you all the private collections as well. If I click on this button now, I can see the collections that exist so far and are publicly viewable. So here's the BICCN Anatomy and Morphology project, for example. And here you can see that I just have access is basically just a portal within the portal that only shows you the studies that have been specifically added to this project. And I can do the same sorts of search functions as I could from the home page, but that will be constrained to this collection. Okay, so I'm going to quickly sign back in just to make sure I can show you all of the features that are accessible to me. I'm going to start out by showing you how to search through these studies. So as I said, there's a simple search option where you just search for a term that might appear in a study description. So I'm going to, for example, look for astrocytes, and this just returns any study with astrocyte in the description. There's also an option to use this more advanced faceted search. So for example, I could constrain my results to studies with data in astrocyte cell types, and then search for something like lung cancer. And here I get a much smaller, more constrained set of data that are easier to sort through. This time I'm just going to go to an individual study, which has been set up just for us, so that we can dig into some data. So when I first click on this study, or any study, I'll see a summary of that study like this. This is the information in the summary is totally up to whoever set up the study or who owns the study, but it often includes a brief overview, a link to a publication if one exists, an abstract, and sometimes there are links also to additional resources. For example, you could link out to visualizations for other portals as well, especially if they're, often these are a really good complement to what visualizations SCP offers. You could also include an image in this description if you want, such as a graphical abstract or a slide image that you is relevant to the data. Then the next tab over is the Explore tab. And this is the real meat of the data exploration capabilities of SCP. So what I have here is just sort of the default plot. So this is a two-dimensional dimensionality reduction. So as I mentioned, the data's original shape is something like number of cells by number of genes. And then that sort of full high dimensional data have been passed through a dimensionality reduction algorithm, in this case a UMAP, to project it into 2D space where it's easier to spot patterns. So every point here corresponds to a cell, and all these points are colored according to the label that's selected in this annotation dropdown. So right now it's selected to display cell type, and you can see from this legend what each of these cell types is. So to make it a little bit easier to identify each of these cell types on this plot, you can just click and then it'll disappear, and click again, and that same group of cells will reappear. It's just sort of like it's waving at you. And so that will help me to sort of easily identify this red set. But it's also, at this point, it's also informative to see things like how many cells belong to each set of each kind of cells. For example, there are a lot of glutamatergic neurons, but not very many macrophages, and also to understand sort of how these are arranged relative to each other. This suggests that the genetic expression profiles for macrophages is pretty different than that for astrocytes. Okay, so now, of course, you can also change this annotation. Instead of cell type, I could color these same data according to donor ID, and notice that everything that colored blue here, which came from one donor, is quite distinct from everything that was in green and red, which came from two other donors. But maybe I have some specific question that bears on that distinction. Okay, so now at this point, you might be wondering what differentiates, I'm just going to go back to the original annotation here of cell type. You might be wondering, okay, so these red and brown cells, for example, seem to belong to two different cell types, but they seem pretty similar to each other in that they're close in this plot. What genetic expression patterns differentiate these two cell types? To try to get at that, you can search for an individual gene up here in this gene search bar. So say I want to look into the P10 gene, I can search for that. And now you'll see a few new visualizations. So the first one I want to draw your attention to is this plot on the left, which is basically the same plot as what's on the right, except now each cell is colored according to the measured P10 expression. And from this, you can see that P10 is kind of expressed throughout the data. There's no one cluster where it's strongest, although you might notice that this sort of serpentine cluster of oligodendrocytes has relatively lower expression. But perhaps this helps to, you might search around for some genes that do distinguish between these cells or these clusters a little bit more. Another way that you can look at this is by clicking over to this distribution tab, which has appeared. And this plots violin plots for, again, P10 expression within each individual cell type. This can be a little bit easier to eyeball sometimes than the plot. You can also play around here with whether you're looking at a box plot or a violin plot and look for outliers, et cetera. There's a lot to explore here. Once you've sort of looked at a P10 expression and are thinking you might be interested in some other genes, you might want to check out this ideogram viewer at the top here. This viewer sort of highlights the gene that you searched for, and then also shows other genes that might be related based on the literature. These are either interacting or paralogous genes, and you can hover over any of these to get a little bit more information. If one of these piques your interest, like if I wanted to look into PTK2, you can just click on that and it will re-render this plot to instead plot PTK2 expression. And here you can see, interestingly, it does seem like this purple cluster of glutamatergic neurons has seemingly at least higher expression of PTK2 than the rest of the data. You have a whole here. Understanding all of these patterns. The last thing that I want to draw your attention to here is that if you found a subset of the data that seems particularly intriguing to you, you can use this create annotation option. This is something you have to be logged in to do, which is why I logged in so I could show it to you. But you basically just click this. You could name that annotation more descriptive than that, hopefully. And then let's say I want to zero in on these macrophage cells. To do that, I just sort of draw a lasso around those cells. I can label these macrophage, not macrophage. Create that annotation. And what happens is that will save that this segmentation of the data. And once it's been saved, which will take a second, you'll be able to see it in this annotations dropdown where you go up to your user menu and then go to my annotations and it'll be there. And then you could download that or share it with collaborators if you want to see what they think of it as well. Okay. Then the last thing on this tab here is if at this point you're intrigued enough by the data that you want to play around with it yourself, you could go around to this, along to this where you can choose individual files to download, or you can click the bulk download button, which will basically just give you a command that you can use in any command line, any terminal window to download all the data at once. One thing to note here is that not all studies will make it possible for you to download your data. Some studies will be private, or they will say something like the data will be released at some future date. And in that case, you might have to email the authors that are associated with that study in order to ask them nicely for their data. So that was a tour through the different ways in which you might explore the data in an individual study. I'm also going to show you how to search for individual genes across studies. So if you go back to the homepage like I just did and click on this search genes option and enter a gene name or multiple gene names, let's go with P10 again, then you'll get some results that basically you'll get a summary of that gene's expression, measured expression across any study that has data on that gene. So for example, here, this one's a little bit more interesting. You see a violin plot that tells you the distribution of P10 expression measured in each of these, in this case, it's clusters of the data, though you could also select some other segmentation of the data, such as the subcluster. This helps you to just sort of quickly eyeball which studies prominently feature a given gene. You can also, as I mentioned, search for multiple genes at once. And when you do that, you will get a slightly different sort of summary. So let's go back down to this study here. So this is a dot plot that tells you, that helps you sort of compare expression of these two genes across different subsets of the data. So for example, here I can see that both of these genes that I searched for are highly expressed in the CA2 cluster of the data, but they are differentially expressed in the GABAergic cluster of the data. And so if I'm looking for differential expression that pulls apart these two genes, this might be an interesting study for me to target. You can also view these plots a little bit differently. You can plot another violin plot where this time you're just averaging across two genes, presumably two genes that you expect to be relatively related to each other. And this will tell you the averaged expression distribution for those genes. Okay, so now I've shown you a few different ways to explore the data. So I'm just going to wrap up in the last few minutes by telling you a little bit of information about how to control access to your study. So I think this is a really key ability that SCP gives you. So I've already created a study of my own. So I'm going to go to my studies. This will take just a second to render because I'm the product manager on SCP, I have access to all the studies, which is kind of a lot. But I'm basically going to open up an individual study that I made. It's just a dummy study. It doesn't have any data. And I'm going to show you how you can share access to your study with individual people, including with anonymous reviewers. So I basically am just going to open up the study, which at this point, as I said, has no data, but this is something that can be done also when it has data, and go over to the settings button. Here, I can change things like the data release date, which I mentioned before. This allows you to make it possible for people to download your data sometime in the future, but not yet. You can also control whether the study is public or private, meaning whether it will show up in search results. And you can share the study with individual people. You can either allow them to view the study, even if it is private, or to edit the study, in which case they could change the data on Terra to sync it with this study. So this is helpful if you have a collaboration going, especially if like one of you is a wet lab scientist and one of you is a computational scientist. You can pass results back and forth. But then the one thing that I think is really useful is this reviewer access. So this makes it possible for you to share access to a study that's still private with an anonymous reviewer so that they can view your data without actually downloading the data. So you would basically just click over to say access is enabled, but if you wanted to disable it at any point, you would just click no. And then an expiration date, meaning when you want the reviewer to no longer have access to your study. And then doing this, once you've made these changes and then clicked update study settings, you can see these reviewer access credentials. So there's a URL and a PIN. You would send these both to your journal editor, essentially, so that they could pass it on to the reviewers without breaking their identity. And so what they would do is once they got this URL, they would click, I should have opened that. Yeah, well, anyway, they would use this URL to access a sort of access point. And at this point, they'd have to enter in their PIN. And once they've entered in the PIN, they'll be able to access your data, but only for 4 hours. This is just to make sure that they're not likely to erroneously share your data with someone else or something like that. If they get kicked out after 4 hours and they still want to go back and see it, they can always just re-enter the PIN. Yeah, so that I took you through a lot of different features there. I know I was hoping to give you a sense of the many different ways that SCP can support your full research life cycle. But I know I just threw a lot of information at you. So if there are any questions, I'd be happy to answer them.
Leyla, I have a question. I realize this might be a silly one, but is there any way to save some of the graphs that you generate in there? Did you did you show that, or?
Yeah, I did not show that, but there is. Yeah, that's a great question. I'm just gonna select this study at random. So if I wanted to save this graph, I can click this camera icon, which will just download the graph automatically as a PNG file.
Okay, very cool. And can you do the same for kind of the dot plots and things that you were showing too? Is that like if you're searching, just across genes? I don't, yeah, like searching across studies or something, and you wanted to save one of those dot plot figures.
I haven't tried to do that, but it might be. I think it has a slightly different control, but you can go to save image. Oh. Yeah, it's a slightly different way of doing it.
Oh, cool.
But that is possible as well. Great question.
Would it be also good to maybe talk a little about the URL and how it can save some of the state of single cell portal?
Right. Yeah. You mean like the URL connected with this particular visualization? Yep. Yeah. So say when I originally landed on this page, I'm gonna go back to. So okay, this is a good demonstration actually. If you've changed something about the visualization, as I have here by searching for a couple of genes, and I want to go back to the original view, I can just click reset view down here. And then, if I wanted to share this exact visualization, say I wanted to change the annotation a little bit, and I wanted to make sure that I was sharing this visualization with a collaborator, I could go over here to get link, and then just copies that link to my clipboard. And then if I were to send that link to someone, they would see the moment of truth. They would see exactly that visualization. So thanks for that reminder, Tim.
I was wondering how many UMaps could you have in here, and how many metadata can you have plotted on the UMaps?
That's a great question. I'm not actually sure of the strict limits there. I mean, like you can see, it depends upon the study how many different UMAP options there are. For example, under clustering here, but you can see if I choose a different one, I actually get a different plot because it's a different projection of the data. Part of the power of SCP is that researchers can choose the different kinds of representations of their data that they want to share. Similarly, sometimes there are just a couple of annotation types, but sometimes they're up to like 20, and it just depends upon what data are supplied. Well, feel free to shoot me an email anytime if you have questions, and I hope you guys get to play around a little bit in SCP.
Thank you so much, Leyla. All right. Well, I'm gonna go ahead and share my screen again. This is Liz. And we can kind of do our little wrap up for today. Before I do that, though, I need to grab a link for you all. Okay. I'm going to be pasting a link into the chat. This is a survey that we would really like you all to just take 5 minutes right now to go answer. And after we come back, after 5 minutes, I'm going to show you some resources, and we can dive into any of the features that you saw today a little bit further or answer any further questions. It's also a good time if you ran into any issues, again, throughout the workshop, trying to do some of the steps, we can talk about that too. So take 5 minutes to fill out that survey, and we'll come back at 3:05, and I will again share some resources with you then. Okay, I hope that was enough time for everyone to fill out the survey. We really appreciate feedback on these surveys. We use them to help, again, tailor the workshops in the future. I'm just going to paste that link to that survey one more time in case anyone wasn't able to fill it out or just didn't see the link that I put there. We really value your feedback. So thank you for taking the time to do that. Can everyone see my screen? Could someone just orally confirm for me that they can see?
Yeah, we can see it.
Thank you so much. Okay, so thank you again for filling out the survey. I wanted to share with you a couple of resources that might be able to help you get started with both Terra and Single Cell Portal. So just starting with Terra, we have some links to different documentation and workspaces that can really help you get up and running quickly if you're interested in moving to the cloud. So starting here, we have some how-to guides on our support forum. We have quick start documentation, understanding data in the cloud, pipelining with workflows, and interactive statistics and visualization with Jupyter Notebooks, as well as Galaxy in our studio. So check out these links. Again, these slides are going to be posted in the workshop folder for the day, and I'll be sending it out by email as well. We also have some great tutorial workspaces. These are workspaces that you can go in and get hands-on with Terra, similar to what you did today. They're exercises that walk you through some really basic concepts of working with different Terra features. And they are designed so that they literally will only cost you pennies to run. So if you have a billing project, it shouldn't use up too much money to run it. And again, you have access to this temp one today, so please feel free to go in and play around if you'd like to play around with any of these. We have ones dedicated to data tables, to our workflows, and also to Jupyter Notebooks. We also have many videos. We have a Terra YouTube channel, which we've linked to here, and you can watch videos that will show people walking through different Terra concepts. There's quick ones, and then there's some longer ones too as well. You'll also find previous webinars on our Terra YouTube channel. So definitely check that out if you're interested in learning more about Terra. And I also wanted to post some single cell resources for you. So I mentioned earlier the Optimus featured workspace that walks you through that 10x data processing pipeline and the overview that lives alongside the GitHub repository. Cumulus also has a featured workspace and overview documentation. And that intro to HCA data on Terra workspace that I mentioned that has the ScanPy notebook in it, as well as a Seurat notebook in it, and Pegasus and Bioconductor. You can check that out if you want to look at some additional Jupyter Notebook examples of working with Loom files. We have SmartSeq2 public workspaces, single cell attack-seq workspaces, single cell methyl-seq workspaces as well, where you can try different pipelines that are also used by that BICCN whole mouse brain working group for uniform analysis. And lastly, we have links for Single Cell Portal. So if you want to go on and play with some of the studies the way based on what Leyla showed you today, you can feel free to do that. And we have a link to the Single Cell Portal documentation. And so with that, I just want to say thank you to you all for joining today. Thank you to Leyla and Anton for speaking and for TAing. And thank you all for your participation. We'll take questions now. And again, we can go back to anything that you would like to play with in Terra and try out different features. If you have any questions at all about migrating to the cloud or how it works, any of the features work, please feel free to ask now. We're a pretty small group, so there's no pressure environment here to get your questions answered. But thank you all so much for your attention today. Any questions?
Hi.
Hi.
I have a question about if you have a sense of how popular the Terra app is going to be or is already. It seems really useful, especially if there's a lot of people depositing their data on it. So I was just wondering if you know, if a lot of people are getting on board with that.
Yeah, so it is definitely increasing. So the nice thing about Terra is it's open source, and again, it's flexible and modular. And the number of people who are using it is very much growing. Terra is already being used for large consortia. So the Human Cell Atlas project is doing all of their data processing now on Terra and storing it in the Terra data repository. We're also, we do processing for the Bro Genomics platform and the Brain Initiative. As I mentioned, some of the working groups are using this for their uniform data processing. So this is all being done. The BICCN, one of the computational environments for the BICCN is the Terra platform. Other large consortia are using it as well, like Anvil and I don't know, Anton, can you think of a few other biodata catalyst? There's several other ones. So it's quite popular, actually. It's becoming quite popular. And I think—
You named the ones I was going to name.
Oh, thanks, Anton. Sorry, I didn't give you time to answer. Yeah, if we think of some more, we can mention it. But and I think, the reason it's becoming used is because there is this pressing need to move to the cloud right now. It makes sense. It is I think most researchers agree that it is the way of the future, because data is just becoming so big, and we want to make use of all of these different data sets that are available from all these different consortia. So hopefully that answers the question.
Yeah, definitely. I have a tangential or related question too. So, people publish like new tools, like if there's a new version of Seurat or something, is that going to get like updated on Terra quickly as well, or?
Yes, so it actually does get updated pretty quickly, quicker than I would like, because I have no has to go in and like check the the work of the notebooks and make sure they work and stuff. And I'm like, oh, like gotta update this. So it does turn over pretty quickly. And the nice thing is you don't have to wait for them to turn over. You can always make I mentioned that we have tutorials on how you can make a Docker image. You can pull in the software. You can update software in real time on Terra. You don't even need to make a new Docker image. You can just update software as you need when you're running those containers. So yeah, it does. It stays up to date, but the whole point of this is that those snapshots of the software, they're always available. So if it's not if you don't want to migrate to the new software that say a Docker container is using, you don't have to. You can keep using the old Docker image. It's all stored in the cloud. They're versioned and they're tagged. So and that's the whole point, is because if you're doing an analysis and you want to share that exact same analysis with your colleagues, you can do that. And you don't have to worry about it, because you're literally giving them the same software dependencies. You're just pulling it from the cloud. It's in a repository in the cloud. So and same with workflows. That's why the workflows are all versioned, so that you can literally give the exact same workflow over. Workflows, the two that I pointed out today, they do turn over quickly. They are updating and pulling in things as people need. So, but you're never limited to using the new version. You will always have access to those older versions of the workflows. So does that answer your question?
Yeah, definitely. Thank you.
Great. Any other questions related to Terra or the cloud or single cell portal?
There's one in the chat.
Oh, there is. Thank you. Let's see, I'm in a BICCN affiliate lab, but we use our local high performance computing clusters. Switching using seems prohibitive when we already pay for access to our university's computing environment. So I don't know, Anton, would you have any feedback on this question in the chat on what to do if you're already in I mean, basically, the short answer is you'd want your institution probably to switch over to a cloud environment ultimately. So if they're already paying for things, anytime someone's already paying for something for you, I'm not going to say you want people to continue paying for things for you, but there are benefits for your institution to move over to create a billing project so that you can run in the cloud, because it scales better.
Right. Yeah, I'm likewise not sure what to say about when your situation involves computing infrastructure that's already paid for. There are advantages to Terra, both in terms of the elastic computation and storage, and in terms of the integration. So kind of building on the earlier question about the popularity, there are the many consortia that are kind of integrating their data with Terra Actually, I wanted to add on top of that, if you go to your profile page, you can see what other entities and infrastructures are integrated with Terra. So like, there's the NIH CRDC, there's Kids First, in addition to the ones that we mentioned like Anvil and Biodata Catalyst. So those are the big advantages. I guess we aren't necessarily the right people to ask about how to compare budgeting between BICCN data analysis, on a.
I guess too, my question for Tricia, yeah, you should bring it up with the BICCN. But my question for Tricia is, how do you, how are you, you said you already pay less for access, but Terra itself, the platform is free and you're only paying for the analysis you do. So I guess I just wanted to know what you mean when you say you're paying less. Are you paying anything? Are you paying zero for that access right now? Or have you done, or do you, like, are you familiar with how much you all are paying for your set infrastructure and how that compares if you were to say, just perform the analysis that you need in a cloud environment? In other words, if you're paying like a finite amount, like let's say $500 a month or a year or something for a certain amount of storage, how does that compare to when you just do the analysis that you need? Is that the kind of the comparison you're making, or?
Hi, well, so I'm a postdoc and I don't actually see any of the breakdown for how much our access to our computing environment costs. But it's essentially we wouldn't pay to run our studio or Surratt on a cloud computing environment when we can do that on our high performance computing cluster where our data is already stored. So, I mean, Terra seems really great for integrating data sets and combining data that way. But at this point, like I wouldn't pay, my PI wouldn't promote or support like paying for the right when I don't remember what it's called, but it was something like 20 cents a minute or something like that.
Oh, no,
right, like that kind of thing.
Yeah, so that's a great point. So you're right, and we don't either for things that we can do locally. There are a lot of, but when you are trying to integrate across kind of cloud data sets and everything, there's benefits to running a virtual computer, in the cloud than doing it locally. And again, it kind of comes down to this ability to sustainably collaborate with people and give them the tools. So I think that's where the benefit comes in. But we always tell people, if you're doing small scale work, you should do that locally. Scale on Terra, do the small stuff locally, because you're right, like if you're just doing a few easy things, you don't want to just kick up a cloud environment to run a simple script that takes you just a little bit to do if it works on your local machine, unless, like I said, collaboration is very key, and it's important that you're sharing with those same resources exactly as you've used them. So but we can find out some more information for this kind of use case where you're working on a high performance cluster, and where the cost benefit analysis comes in, because there are people who know more than me and Anton on this, I think, right now. So we can follow up with you if that would be good and see what we can find out.
I did want to add that so in the same vein as collaboration, like it's a really great tool for collaborating, if you have collaborators outside of your university, but also it's a great way to publish things or to put things up publicly. So you can create featured workspaces of your own, for example, in order to share with the general public. So once you're ready to, integrate your results with the cloud for the scientific community. Oh
yeah, absolutely. I think that the BICCN is having all of our data submitted to NeMO. Yeah, I guess maybe I'm just a little unsure of like when we should be using these different tools, right? But I think that's just a larger question about that isn't really necessary. It's not, you guys aren't really like the people that I should be asking that question of.
It's a great question, though. It really is. Like, when should you make the move to the cloud? Like, when does it become necessity? And so, yeah, it's a great question. I'm sorry we can't give you more feedback on it.
No, something tells me that there's probably not an answer to this yet.
All right. Well, I think we're all happy to stick around and answer any questions. But you can, feel free to, to go enjoy the rest of your day. We really appreciate your attendance today and participation.